Hi, my name is Shahar. I'm from Sagol School, Tel Aviv University, and my project is about elucidating cyclic AP dynamics in the mossy fiber synapse using novel optogenetic and imaging tools. Synaptic plasticity is thought to be one of the main cellular processes that underlies learning and memory. It is implicated in several synapses, especially the hippocampal mossy fibers, that a rise in presynaptic cyclic MP is a crucial component in elevating neurotransmitter during mossy fibers LTP, long-term potentiation. So far, cyclic MP-dependent synaptic process was obtained by using pharmacological agents, but their effect is not specific nor reversible. In order to overcome this obstacle, we use BPAC, a photoactivatable adenylyl cyclase from a marine bacterium, which mediates light-dependent cyclic MP increase. Its clear advantage is its ability to express its fast and reversible activation in a cell-specific manner. In addition, we use pink flamingo, a cyclic MP biosensor, to track cyclic MP concentrations and dynamics in neurons following BPAC activation. So, how it is done? Our experiment goes like that. We have our presynaptic part of the mossy fibers and the postsynaptic part. We have BPAC, our cyclic MP elevator, and pink flamingo, our sensor. We activate BPAC with blue light illumination and then cyclic MP elevated at the cell. Cyclic MP binds to the binding domain of the pink flamingo, and as a response, pink flamingo emits red light. Hopefully at the end, synaptic transmission occurs. But first of all, in order to measure cyclic MP dynamics, we basically use our cyclic MP sensors. I took pink flamingo in an AUV construct and transduced neurons with it. This graph shows the normalized fluorescent intensity of pink flamingo through time. When I applied a cell-permeable cyclic MP analog, I got an increase in fluorescent intensity, which means that pink flamingo was capable to track elevation of cyclic MP levels at the cell. Next, we tried to do an in vitro cyclic MP calibration in HEK cells in order to determine absolute levels of cyclic MP using pink flamingo, our sensor. We have applied the same cyclic MP analog in live HEK cells in red and pure cyclic MP in a lysate of HEK cells in green. We can see that while we increase cyclic MP, we get a higher fluorescent intensity of pink flamingo. And finally, my main experiment. In this experiment, I checked for cyclic MP dynamics following BPAC activation in neurons. I co-transduced neurons with pink flamingo and BPAC in an AV construct. I activated BPAC with variable light pulse duration. In the blue graph, we can see that when I expanded the light pulse duration, I got an increase in fluorescent intensity, which means more cyclic MP in the cell. We can see it right over here in this footage. In the pink graph, we can see that when we have no BPAC in the cell, we don't get the response of cyclic MP elevation, which means no BPAC, no response. Now we're examining cyclic MP dynamics following different stimulation protocols like LTP, long-term potentiation, and LTD, long-term depression. Meanwhile, we are developing an experimental system that will allow us to activate BPAC only in the presynaptic part of the mossy fibers. These tools will help us monitor cyclic MP in a cell-specific manner. Oh, and also, the connection between cyclic MP and mossy fibers plasticity. Do you want to hear more?